Let us start with general parasitology and the overview of parasitic infections. As this is a very important yet neglected unit, so parasitology as well as mycology will be available as separate playlist as well, and at the same time they will be available with this respective units as well. Okay, in the playlist, please check. Now, in this chapter, we will be previewing general parasitology, life cycle of parasites, lab diagnosis and treatment of parasitic diseases, overview of parasitic infection, protozoan infection, helminthic infection. In this chapter, in this video, we will be covering the general parasitology, life cycle of parasites, and the lab diagnosis, while the further parts will be covered in the subsequent videos. General parasitology. Parasite is a living organism which lives in or upon other organisms and derives nutrition directly from it without giving any benefit to the host. Medical parasitology deals with the study of animal parasites which infect and produce diseases in human beings. Parasites may be classified as ectoparasites. They inhabit the surface of the body of the host without penetrating into the tissues. Example: fleas, mites, or ticks. They serve as important vectors transmitting the pathogenic microbes. The infection produced by these parasites is called as infestation. Example: scabies. Endoparasites, these are the parasites that live within the body of the host. Example: leishmania. Invasion by the endoparasite is called as infection. The endoparasites are further classified into protozoans and helminths. Protozoa, they are the unicellular eukaryotic cells that perform all the physiological functions. Although like bacteria they are unicellular, they are considered as low eukaryotes as they possess cellular organelles and metabolic pathways similar to that of eukaryotes. Helminths, they are elongated, flat or round worm-like parasites measuring few millimeters to as long as few meters. They are eukaryotic, multicellular and bilaterally symmetrical. Medically important parasites we'll see in the table. Okay, now the life cycle of parasite. The life cycle of parasite depends upon three factors: host, mode of transmission and the infected form. Host is an organism which harbors the parasite and provides the nourishment and shelter. Host can be classified into definitive host, where the parasite undergoes the sexual cycle, or the intermediate host, where the parasite undergoes asexual cycle. Now, in this table, let us see some medically important protozoans and helminths. Medically important protozoans: amoeba and Amoeba histolytica, free-living amoebae like Naegleria floweri, Acanthamoeba, and Balamuthia. Flagellates like intestinal flagellate Giardia, genital flagellate Trichomonas, hemoflagellate Leishmania and Trypanosoma. Epicomplexa, malaria parasites and Babesia. Opportunistic coccidian parasites Toxoplasma, Cryptosporidium, Cyclospora, Cystoisospora and Sacrocystis. Miscellaneous protozoa like Balantidium coli, Blastocystis and Microsporidia. Now the medically important helminths are cestodes like Diphyllobothrium, Taenia, Echinococcus, Hymenoplepis and Dipylidium and others. Trematodes or flukes like Schistosoma, Fasciola, Clonorchis, Ophisthorchis, Phaseolopsis, Paragonimus and others. Intestinal nematodes like Trichuris, Enterobius, hookworm, Strongyloides, Ascaris and others, and somatic nematodes like filarial nematode, Ranunculus and Trichinella. Microsporidia are now considered to be evolved from fungi. Okay, and ectoparasite infestation we will discuss later. Now, depending upon the number of hosts involved, the life cycle of parasite may be direct, simple, or indirect, complex. The direct or simple cycle, the parasite requires only one host to complete its development. In indirect or complex life cycle, parasite requires two or three hosts, one definitive and another intermediate host to complete the development. Infective form, it is morphological form of the parasite which is transmitted to man. Now, the life cycle of various protozoan parasites. Entamoeba and Giardia, definitive host man, mode of transmission ingestion, infective form cyst, polynucleated cyst. Trichomonas vaginalis, man, mode of transmission sexual, infective form trophozoites. Leishmania species, definitive host man, intermediate sandfly, mode of transmission vector-borne, infective form promastigotes. Trypanosoma cruzi, definitive host man, intermediate host radiovid bugs, mode of transmission vector-borne, infective form trypanomastigotes. Trypanosoma buceri, definitive host man, intermediate host CC fly, mode of transmission vector borne, infective form trypanomastigotes. Plasmodium species, definitive host Anopheles, intermediate host man, mode of transmission vector borne, infective form sporozoites. Toxoplasma gondii, definitive host cat, intermediate host man, mode of transmission ingestion, blood transfusion vertical, infective form tissue cyst, oocyst, and tracheozoids. Cryptosporidium, definitive host man, no intermediate host, mode of transmission ingestion, autoinfection, infective form oocyst, sporulated. Cyclospora cystoisospora, definitive host man, mode of transmission ingestion, infective form oocyst, sporulated. Now, the life cycle of various helminthic parasites. Cestodes, Taenia solium, intestinal teniasis, definitive host man, intermediate host pig, mode of transmission ingestion, infective form Cysticercus larvae. Taenia solium cysticercosis, definitive host man, intermediate host also man, mode of transmission ingestion, autoinfection, infective form embryonated eggs. Cestodes, Taenia saginata, definitive host man, intermediate host cattle, mode of transmission ingestion, infective form Cysticercus larvae. Echinococcus granulosus, definitive host dog, intermediate host man, mode of transmission ingestion, infective form embryonated eggs. Hymenolapis nana, definitive host man, no intermediate host, mode of transmission ingestion, autoinfection, infective form embryonated eggs. Typhlobothrium lantum, definitive host man, intermediate host first cyclops, second fish, mode of transmission ingestion, infective form plecocercoid larvae. Now, trematode. Schistosoma species, definitive host man, intermediate host snail, mode of transmission skin penetration, larva, infective form is Cercaria larva. Fasciola hepatica, Fasciolopsis buscae, definitive host man, intermediate host snail and aquatic plants, mode of transmission ingestion, larva is Metacercaria larva. Paragnomius species, definitive host man, intermediate host snail and crab, mode of transmission ingestion, infective form Metacercaria larva. Cloarchus or Ophisthorchis species, definitive host man, intermediate host snail and fish, mode of transmission ingestion, larva is the Metacercaria larva. Now, intestinal nematodes, Ascaris trichuris, definitive host man, mode of transmission ingestion, infective form is embryonated eggs. Enterobius species, definitive host man, mode of transmission ingestion, autoinfection, uh, infective form is embryonated eggs again. Hookworm infection, definitive host man, skin penetration is the mode of transmission, and L3 filariform larvae is the infective form. Strongyloides stercoralis, definitive host man, mode of transmission skin penetration, autoinfection, infective form L3 filariform larvae. Now, lastly, somatic nematodes, filarial worms, definitive host man, intermediate host mosquito and flies. Transmission is vector borne and infective form is L3 filariform larvae. Dranunculus mediensis, definitive host man, intermediate host cyclops, mode of transmission ingestion, and infective form is L3 larvae. Trichinella spiralis, definitive host man or pig, mode of transmission ingestion, and infective form is L1 larvae. Those which are star marked, and those man acts as accidental intermediate host for Echinococcus granulosus. Okay, now we get it. Now let us come on to mode of transmission. Parasites may be transmitted by various modes such as ingestion, skin penetration, vector-borne, sexual, vertical, blood transfusion, and autoinfection. Autoinfection. Few intestinal parasites may infect the same person by contaminated hand, external autoinfection, or by reverse peristalsis, internal autoinfection. It is observed in some parasitic infections such as Cryptosporidium parvum, Taenia solium, Enterobius, Vermicularis, Strongyloides pterocoralis, and Hymenolopsis nana. Now, coming on to lab diagnosis of parasitic diseases. Lab diagnosis plays an important role in establishing the specific diagnosis of various parasitic infections. Following are the techniques used in the diagnosis of parasitic infections. Examination of feces. As many parasites inhabit the intestinal tract, stool examination is the most common diagnostic technique used for the diagnosis of parasitic infections. Specimen collection. Stool specimens should be collected in a wide-mouthed, clean, leak-proof, screw-capped container and should be handled carefully to avoid acquiring infection from the organism present in the stool. Timing. Specimens should be collected before starting the antiparasitic drugs and closer to the onset of symptoms. Frequency. At least 3 stool specimens collected on alternate days within 10 days are adequate to make the diagnosis of intestinal parasitic diseases, except for intestinal amoebiasis, for which 6 specimens may be recommended. And to examine, liquid stool specimens should be examined within 30 minutes, semi-solid stool within 1 hour, as on storage trophozoites may disintegrate or become non-motile and form stools up to 24 hours after collection. For monitoring response to therapy, repeat stool examination can be done 3 to 4 weeks after the therapy for intestinal protozoan infection and 5 to 6 weeks for tinea infection. If delayed in transport, fecal specimens should be kept at room temperature. Preservative, example 10% formalin, can be used to maintain the morphology of the parasitic cyst and eggs. Specimens other than stool, like perianal swab, cellophane tape, or NIH swab, useful for detecting eggs of Enterobius vermicularis deposited on the surface of perianal skin. It is also used for eggs of Schistosoma mansoni and Tinea species. The ordinal contains very useful for the detection of small intestine parasites like Giardia intestinalis and larvae of Strongyloides stercoralis. Duodenal fluid may be collected by endoscopy or by enterotest. Microscopic examination. Microscopic examination of stool may provide clue about various parasitic infections. Mucoid bloody stool found in acute amoebic dysentery, intestinal schistosomiasis, and invasive bacillenteriasis. This is the sample container for stool, okay? Now, color dark red stool indicates upper gastrointestinal tract bleeding, and a bright red stool is suggestive of bleeding from the lower GIT. Frothy pale offensive stool containing fat is usually found in giardiasis. Stool consistency. In liquid stool, trophozoites are usually found, whereas in semi-formed stool, both trophozoite and cyst are found, and the cysts are mainly found in formed specimens. Exceptions to this general statement include coccidian oocytes, microsporidian spores, helminths, eggs that can be found in any type of fecal specimen, and cryptosporidiosis, oocytes load is higher than liquid stool. Tapeworm, troglodytes, an adult worm of Enterobius and Ascaris are occasionally found in the stool. Microscopic examination. Microscopic examination includes direct wet mount examination and permanent staining methods. Direct wet mount, saline and iodine mount. Drops of saline and Lugol's iodine are placed on left and right halves of the slide respectively. A small amount of feces, approximately 2 mg, is mixed with a stick to form uniform smooth suspension. If more or less fecal matter has been taken for the stool wet mount, the chance of finding stool parasites decreases. In this diagram, we see the comparative availability of cyst and trophozoites. Like as we go from firm to soft to loose to watery stools, the number of cysts decreases while trophozoites increases. Okay, and this is the saline iodine wet mount of stool examination. This is the method of screening of the slide during wet mount examination of stool. Cover slip is placed on the mount and examined under low power objective 10x for detection of helminths, eggs and larvae, followed by high power objective 40x for protozoan, cyst and trophozoites. Okay. Now screening area, the entire cover slip preparation should be examined in a zigzag fashion, first under low power and then under high power objective before reporting a negative result. Motility. If the finding is suspected to be a trophozoite, then examination for at least 15 seconds should be allowed to detect motility. Motility can be stimulated by application of heat by placing a hot penny on the edge of a slide or tapping the cover slip or increasing the intensity of the light source. The following are the structures that can be visualized by microscopic examination of the stool specimen, which may be confused with various protozoan trophozoites, cyst or helminthic eggs and larvae. Normal constituents, these include plant fibers, starch cells, stained blue-black with iodine, muscle fiber, animal hair, pollen grain, yeast cell, bacteria, epithelial cell, fat globules and air bubbles, etc. Now in these figures, these are important egg figures, okay. In A and B, we see the yeast cell resembling like A is Giardia cyst, B is Cryptosporidium oocyst. Okay, C2E, these are fungal spores resembling. C is the cyst of entamoeba. D and E are cryptospora oocyst. Okay, now F to K are pollen grain resembling. F is blastocystis, it is trichome staining. G is blastocystis, saline mound. H is protozoan trophozoite. I is ascaris egg. J is clonorchis egg. K is tinea egg. L and M are plant cell resembling. L is helminth egg and M is hookworm egg. N is plant hair resembling strongyloides larvae. O is diatoms. P is mite egg resembling hookworm egg. Q to S are crystals. Q and R are pineapple juice crystals and kiwi crystals. S is charcoal laden crystals. T to W, human cell resembling trophozoites. T is white blood cells. U are macrophages. V and W are epithelial cells. X are air bubbles and Y are fat globules. Now the cellular elements like pus cells in inflammatory diarrhea, red blood cells in dysentery may be present. Charcoal laden crystals diamond shaped, they are the breakdown products of eosinophils and may be seen in the stool or sputum of patients with parasitic diseases such as amoebic dysentery, ascariasis and allergic disease like bronchial asthma sputum. Saline mound is useful in the detection of trophozoites and cysts of protozoan parasites and eggs and larvae of the helminths. It has the following advantages than iodine mound. Motility of trophozoites and larvae in acute infection can be demonstrated. Bile staining property can be appreciated. Bile stained eggs appear golden brown and non-bile stained eggs appear colorless. In stool specimen with preservative, directly the wet mount can be prepared without using saline. Iodine mount. Advantages: nuclear details of protozoan cyst, helminthic eggs, and larvae are better visualized compared to saline mount. Disadvantages are iodine immobilizes and kills the parasites, hence motility of trophozoites and helminthic larvae cannot be appreciated. Bile staining property cannot be appreciated. Non-bile stained eggs. Eggs of most of the intestinal parasites when they pass through the intestine are stained by bile, the exceptions being Enterobius, hookworm, and Hymenopsis nana. These eggs are non-bile stained. Permanent stain smear. Permanent stain smear are required for accurate detection of protozoan cysts and trophozoites by staining the internal structures. Commonly used methods are iron hematoxylin stain, trichome stain. Modified acid fast stain. This is useful for coccidian parasites such as Cryptosporidium, Cyclospora, and Cystoisospora. Now the concentration techniques. If the parasite output is low in feces, example cysts, trophozoites and larvae, and direct examination may not be able to detect the parasite, then the stool specimen need to be concentrated. These methods are also useful in epidemiological analysis and for assessing the treatment response. The egg, cyst and larvae are recovered after the concentration procedures, however the trophozoites get destroyed. Commonly used concentration techniques are sedimentation techniques. The eggs and cysts settle down at the bottom of the tube because they have a greater density than the suspending medium following centrifugation. Example include formalin ether concentration technique. The sensitivity of the detecting ova or cyst increases by 8 to 10 folds. The size and shape of the parasitic structures are maintained. Now the flotation technique, it involves suspending the specimen in a medium, example saturated salt solution of greater density so that the helminthic eggs and protozoan cyst float on the surface of the solution. The disadvantage is it cannot be used for the concentration of the parasites that do not float in saturated salt solution such as unfertilized eggs of Ascaris, larvae of Strongyloides, tinea eggs and opiculated eggs of trematodes. Other flotation methods are zinc sulfate flotation concentration technique, Sheether's sugar flotation technique useful for Cryptosporidium, Cystoisospora and Cyclospora. Various morphological forms of parasites seen in the stool specimen are listed in the table. Now, egg counting, egg quantification method. The intensity of the intestinal helminthic infection, especially trichoris, ascaris, and hookworm, can be estimated by egg counting in the feces, which can be performed by various egg counting methods. Direct smear counting method of Bavier, Cato-Katz thick film method, Stoll's method, or dilution egg counting method. Now, examination of blood. Blood examination is useful in the diagnosis of infection caused by blood parasites like Plasmodium, Trypanosoma, Leishmania, Babesia, Wucheria, Bancrofti, Brugia malai, Loa Loa, and Manzanella. Morphological forms of parasites seen in two specimens. Trophozoite and cyst are seen in Entamoeba histolytica and Giardia lamblia. Adult form is seen in Ascaris lumbricoides and Enterobius vermicularis. Adult worm segments are seen in Tinea species and Phyllobothrium latum. Egg is seen in Diphyllobothrium latum, Taenia species, Hymenopsis nana, Schistosoma species, Fasciola hepatica, Fasciolopsis buscae, Ascaris lumbricoides, hookworm, Enterobius vermicularis, Trichuris, Trichura, and larvae seen in Strongyloides turcoralis. Now coming on to various methods of examination of the blood. Direct wet mount examination useful for detection of malaria parasite and microfilariae and lymphatic filariasis. Examination of blood smears, thick smear and thin smear and thick smear are examined after staining with various Romanovsky stains such as Leishman stain, Jimsa stain, Field stain, and Jaswant Singh and Bhattacharjee JSB stain. This is the most common method of microscopic examination of peripheral blood useful for most of the blood parasites. Quantitative Buffy coat QBC involves collection of the blood in a capillary tube coated internally with acridine orange stain, centrifugation and then examination of the Buffy coat region under fluorescent microscopy. This is extremely useful for the detection of the malaria parasites and microfilariae. Concentration of blood is useful for detection of microfilariae from the blood specimen. Various concentration methods are sedimentation technique, cytocentrifugation, cytospin, knot concentration, gradient centrifugation, and membrane filtration. Microscopic examination of other specimens. Microscopic examination of various specimens other than stool can also be performed and demonstrated to demonstrate the different morphological forms of the parasites. Now, the immunodiagnostic methods involve detection of parasite-specific antibodies in serum and detection of circulating parasitic antigen in the serum. These methods are useful when parasites are detected only during the early stage of the disease, parasites occur in very small numbers, parasites reside in the internal organs, and morphological identification is not possible when other techniques like culture are time consuming. Antibody detection test. Antibodies are detected in various parasitic infections, mainly from serum, sometimes from other specimens such as CSF, neurocysticercosis, or pleural fluid paragnomiasis. The parasitic disease where antibody detection methods are useful are amoebic liver abscess ELISA detecting antibodies in serum against 170 kilodalton of lectin antigen, visceral leishmaniasis detecting antibodies in serum to RK39 antigen by immunochromatographic test ICT, toxoplasmosis Sabin Fledman dye test, a complement mediated neutralization test which detects antibodies, detection of specific IgM, IgA or IgG antibodies by ELISA. Cysticercosis ELISA detecting antibodies against purified glycoprotein antigens, Western blot detecting antibodies against highly specific 50 to 13 kilodalton lentil lectin purified C7 glycoprotein LLGP antigenic fractions. Hydrated disease ELISA detecting antibodies against B2T or 2B2T antigen and DIGFA dot immunogold filtration assay. Lymphatic filariasis flow through assay detecting antibodies against recombinant filarial antigen WBSXP1 and Brugia rapid detecting antibodies against recombinant B malai antigen BM14. Okay, now in this table we can see the morphological forms of parasites seen in different specimens other than stool. In peripheral blood smear, ring form, schizont and gametocyte is seen in Plasmodium species. Amastigote is seen in Leishmania species. Trypomastigote is seen in Trypanosoma species. Microfilaria is seen in filarial nematodes. In bone marrow, liver, lymph node and splenic aspirate, trichizoid is seen in Toxoplasma gondii. Amastigote is seen in Leishmania donovani. In liver aspirate, trophozoite is seen in Entamoeba histolytica. Lymph node aspirate, trypomastigote is seen in Trypanosoma species. In lymph node biopsy, adult worm is seen in Bucheria bancrofti and Brugia malai. In CSF, trophozoite is seen in Naegleria floweri and Acanthamoeba species, and trypomastigote is seen in Trypanosoma species. In urine, trophozoite is seen in Trichomonas vaginalis, microfilaria is seen in Bucheria bancrofti, and egg is seen in Schistosoma hematobium. In sputum, adult worm is seen in Paragnomius species, egg is seen in Paragnomius species, and larva migrating is seen as Ascaris lumbricoides and Strongyloides species hookworm. And trophozoite is seen in Entamoeba histolytica. In duodenal aspirate, trophozoite is seen in Giardia lamblia and larva is seen in Strongyloides stercoralis. In corneal scraping, Strophozoite is seen in Acanthamoeba species. In skin, Emastigote is seen in Leishmania species, Microfilaria seen in Onchocerca volvulus, and larva in skin ulcer fluid is seen in Ranunculus mediensis. About muscle tissue, encysted larva is seen in Trichinalia spiralis, and cysticercus cellulose is seen in Tinea solium, while in perianal area, egg is seen in Enterobius species. Filial nematodes found in the peripheral blood smears are Bucheria bancrofti, Rogia malai, Loa loa, and Mansonella species. Now, let us come on to the antigen detection test. Antigen detection methods are available for various parasitic disease. Amoebiasis, ELISA detecting 170 kilodalton of lectin antigen in blood and stool. Triage parasite panel, it is ICT that simultaneously detects 3 antigen in the stool. Giardia alpha 1 giardin antigen, E. histolytic or E. dispar 29 kilodalton antigen, and Cryptosporidium isomerase antigen. Malaria ICT format available detecting histidine rich protein 2 PF HRP2 Plasmodium falciparum specific, parasite lactate dehydrogenase PLDH and aldolase common to all species. Lymphatic filariasis ELISA and ICT formats are available detecting filarial antigens by using monoclonal antibodies against OG4C3 and AD12 antigens. Molecular methods most frequently used in diagnostic parasitology include polymerase chain reaction PCR and real-time PCR have been developed for most of the parasitic infections. LAMP assay has been developed for visceral leishmaniasis and malaria. Biofire film assay is an automated multiplex nested PCR. The gastrointestinal panel can simultaneously detect 22 enteric pathogens including 4 parasites: Entamoeba histolytica, Giardia lamblia, Cryptosporidium and Cyclospora. Now, other diagnostic modalities, culture techniques. The culture techniques have been described for various parasites such as amoeba, trichomonas, malaria parasite, leishmania, and nematode larvae. They are not routinely done in diagnostic parasitology but useful in research and teaching purposes. Imaging techniques. Being non-invasive methods, imaging techniques such as X-ray, ultrasound, computed tomography, CT, and magnetic resonance imaging (MRI) are extensively used for various space-occupying parasitic infections such as amoebic liver abscess, cysticercosis, and hydrated disease. Intradermal skin tests. Positive intradermal skin tests are suggestive of past exposure as they remain positive for longer duration. They cannot differentiate between the old and recent infections. Skin tests showing immediate hypersensitivity are available for hydrated disease, Cassoni's test, filariasis, schistosomiasis, ascariasis, strongyloidiasis, and trichinellosis, Bachmann test. Okay, now skin test showing delayed hypersensitivity available for leishmaniasis, Montenegro test, trypanosomiasis, and toxoplasmosis. Xenodiagnostic technique. Here the arthropod vectors are fed on infected patients 2 to 4 weeks, following which the vectors are examined for presence of parasites. The test is seldom used for diagnosis of Chagas disease. Okay, animal inoculation methods mainly useful for research purpose, not routinely used in diagnosis of parasitic infection. The animal models are specific for different parasites. For example, mice for toxoplasma, trypanosoma hamsters for leishmania, and rats for toxoplasma, trypanosoma, and trichinella. Okay, so this covers part 1 of general parasitology. Next part will be covered in the next video. Okay, like, share, subscribe. Thanks for watching.